Hello everyone, I am Faraz Siddiqui, representing Biofootprints, a society which aims at science promotion and popularization. Today we have a special guest with us, who is no less than a milestone in the field he steps in. His innovations and inventions are just a tiny show of what he really is, nothing less than excellence and a big image of motivation and hard work. So with this, I would like to introduce Professor Asad U Khan, and a very warm welcome, sir. It's a privilege to have you on this platform. So, a little introduction of Professor Khan. He has graduated in chemistry and did his postgraduate in biotechnology and obtained a doctorate in biochemistry from Aligarh Muslim University, and later proceeded for his role doctor in RUTGERS University New Jersey, USA during 2000 and 2003 for 3 years. Currently, he is professor and ex-coordinator Biotechnology Unit, AMU, Aligarh, India. He has been involved in teaching microbiology at postgraduate and doctoral level for last 23 years and has guided 25 PhDs, 17 MDs, MPhil, and 54 master dissertations in the area of antibiotic resistance and therapy. He has published over 240 research articles in the area of antimicrobial resistance mechanism in peer-reviewed high-impact journals with H-index 41, which has been recognized in the scientific and public domain. His work has been cited in Nature, Science, Cell, EMBO J, JBC, etc. His research findings on antimicrobial resistance have been covered by both international electronic and print media and twice broadcasted by French television. Elected Fellow Royal Society Chemistry, recipient of INSA-International Collaborating Bilateral Exchange Programme, Fellowship of Biotech Research Society, India, Fellow of Indian Academy of Microbiological Sciences, recipient of National Bioscience Award for Career Development of Government of India, DBT, first recipient of Outstanding Research Award of the Year conferred by AMU, recipient of Wockkardt Excellence Award of 2018, and recipient of Visitors Award of President of India in 2019. The list is never ending, sir. And with this, I would conclude and invite you to start with your presentation. And I'm sure there'll be a lot of people who would like to hear your journey in your words. Over to you, sir.
Thank you, Faraz, for a very nice introduction, and I thanks also to this YouTube for Biofootprints, who's doing good service for encouraging the youngsters to inspire peoples. Well, so I've been given a chance to talk on my journey in the field of my research and science. So I'll just make this talk not more of scientific for a specific area, for a specific audience, but I made it, the way I can communicate how one can proceed and excel in the area of their own research and own scientific area. So that's what I have made this presentation. So I'll start just from my talk, from my starting point as a PhD. I've been registered in Aligarh Muslim University back in 1994 after completing my PhD, and then I got a chance as the first Indian researcher work in this ICGB, because at that time in 94 ICGB was meant for international students, but I was the first got chance to work with Dr. Sunil K. Lal. Professor Sunil Lal was the newly joined ICGB from Caltech, and he guided me a lot, and the whole work was carried out in ICGB in collaboration. Definitely part of the work was done in Aligarh Muslim University. I was registered in Professor Masood Ahmad, who was a professor in biochemistry, and finally we submitted the thesis on the very interesting topic at the time. It was a T4 splicing. So I started working on RNA splicing of T4 phage, which is the bacterial system, prokaryotic system, very interesting. And my main focus is on ribosomal RNA of it, is self-splicing RNA. And I focus on NRDB gene, which is ribonucleoreductase gene, and that I work on looking the functional aspect of this intron and expression of a gene. This is just a glimpse of some slides. I'm not talking the whole data. Just this is the basic mechanism for learner, how does splicing works, and what are the basic principles behind the splicing. So what we have worked on, looking certain aspects of defect in splicing using different mutants. So we have generated at least more than a dozen of mutants, laboratory mutants, and these mutants were later on confirmed with the defect in a splicing mechanism of the different ants. So this is just the work we published in a respectable journal in 1998. This was my first paper I published in 98. BBRC, then again 2000 we had BBRC, and then Journal of Biomedical Sciences in 2003. After that we joined Aligarh Muslim University as an assistant professor. Even before I defended my thesis in 1998, I joined in 97, and I tried to establish a lab focusing my own research area in Aligarh Muslim University in the biotechnology department we have. Now, but due to some limitation of my work, limitation of some infrastructures, I thought of rather than establishing here to take a lead to move abroad. And I got a postdoc position in RUTGERS in Robert Wood Johnson Medical School, one of the leading school in New Jersey. There, I worked with Professor Michael Hamsey, who is a transcription biologist. So right from RNA, I moved to more of a splicing and moved to the transcription biology. And I worked on very interesting model of functional interaction between RNA polymerase II in 3' end processing. So what I did, we tried to connect, and that study was published in Genes and Development in 2002. What I did is basically we tried to connect the SSU72 gene with the TF2B basal transcription mechanism of the poly2 transcribed gene. And what I did, if you look into the role of SSU72 in the initiation complex and then leading to the termination complex, that's how the connection made. So what we did, we connect the dot between the initiation and termination in the E system. And that was the first kind of a study published in Genes and Development. Later on, several other factors were discovered in the same lab after I left. But I think the whole model was completed with the SSU72 gene would connect the with the endonuclease activity and other activities. Well, if you look at the transcription model, this is the first factor initiation complex of transcription. So what we have shown first time that SSU72 was associated with the CPF cleavage cleavage factors of the which is involved later on in the cleavage vectors. Now, I'm not going to talk about the detail of it, but what is more important here is that and more fascinating that I have shown the role of SSU72 in the FCP, where the transcription initiation starts. So what we have proved that SSU72 was involved in dephosphorylation as a putative activity of the phosphatase and dephosphorylation of Hall2 CTD domain, and that was very important concept now. And even if you go to the books, you find these references. Well, this is my group, very interesting group. Remember, I'm connecting with several. This is Shankar Ling was joined later to me, and then Michael Hamsey, my boss, and these are two members of the group, graduate students and some undergraduate students. Now, the focus of my research, I'm back from US in 2004, back in India, and resume a position again as assistant professor, and I focus on basically on antimicrobial resistance. Because that was a not very hot cake at that time, but definitely now it is a hot cake in the last 20 years. And my focus is on mechanism, structure, biofilms, using different approaches like genomics, proteomics, bioinformatics, gene editing, and CRISPR-Cas system. I'm also working on therapeutic aspect of it by inhibitory effect of several molecules using as a designer molecule, I call them, or engineered molecule, I call them, and PDT nanomedicine. Now, well, having that in idea, I thought of initiating several projects, and I've been given several grants from different funding agencies, DBT, CSIR, and others. We started the project in the core shell engineered nanomaterials to target the infection model. So we developed several infection models to handle the situation of the AMR in the absence of antibiotics. We also work on metagenomics, on pan-DMR, CRISPR-Cas is another project I'm working. Now, then I moved to 2005. After working a year almost, I got a project and then moved to Italy on DST Bioscience Fellowship and got a chance to work to another stalwart in field of antimicrobial resistance, Professor Raffaele Zerilli in Nepal. He's a man of internal medicine. And there we learned a lot of tools of AMR because during my PhD and my postdoc, I am not trained in AMR, though we have trained in basic techniques like molecular biology, cloning, gene expression, knockout, knockdown, but the model I've chosen here in India as AMR, and we learned a lot of techniques in the area of AMR in the Lello's lab, and then we got published in 3 months. We got one good papers in AAC in 2008. That paper came quite later. Well, this is my group in Italy. I seems to be very young. almost 15 years ago. And this is the nice Italy and Nepal area. Now, coming back again in 2005 onward, I started working on the project, same inhibitor designing of metallo-beta-lactamases. Then I work on biofilm. Then I work on, proteomics to understand the whole pathways in biofilms, and working on infection control models by DFU and chronic infections and the metagenomics of Pan-India. So these are my even current students, those are working in my lab, working in the different projects. Then 2010, I moved to, under the DBT-CREST program of Government of India, I got a chance to go, one of the top lab in AMR is Patrice Nordman. He's the man behind the NDM1 discovery. So Timothy Walsh and Patrice Nordman are the pioneer in the field of AMR, and their work, basically he identified the NDM. So when I joined his lab in 2010, October, probably July, then he had a joke with me in a cafeteria in the first meeting that he said, people are jealous to me because they said they have given the name NDM because his name is Patrice Nordman. So he is correlating his name with the Patrice Nordman with the NDM, but it's not the case. Case is that it is a New Delhi metallo-beta-lactamases. There's a lot of controversies goes on in that naming the NDM, but he's the one who defended it, and finally the NDM one was identified. So Patrice Nordman is a stalwart and is one of the pioneer area of AMR, and we got a chance to work and learn a lot of other techniques before I established my group in India. And we got a chance to publish 2 papers with his lab in, again, 3 months' time. One is Journal of Medical Microbiology and Scandinavian Journal of Infectious Diseases, where we have shown how NDM in Enterobacteriaceae and cloacae, Klebsiella pneumonia, work in a diabetic foot ulcer in India. And that's how the same strains were used in my later studies, where we try to control the infection, diabetic foot ulcer infection, in the later stage by using different technology. Well, now my lab is fully established in the field of AMR after having this much experience in a worldwide experience. So I first started working on one of the based on my biochemistry knowledge, the mechanism of antimicrobial resistance to understand the structure inside using the site-directed mutagenesis. So I have generated like a dozen of mutants, though it's very difficult to make NDM mutants and CTX mutants, which are the core markers of the AMR, but we did it. And thanks to my students who is now in USA, in Pittsburgh, who was working in one of the labs where in cancer biology, looking the protein expression part, which he learned from my lab. And we have produced like a dozen of a protein which have been expressed from different mutants we generated, and we express the proteins, and we did a lot of studies on that, and we try to map the structural part, different region of these NDM, which are directly involved in binding of several drug molecules. We also try to overlap homology modeling of the several sequences of variants of these NDM, and try to understand what are the conserved residues involved in the interaction of the drug, and so we can design inhibitors. And that's a long work. We started in 17, 18, 19, 20, and we got good papers out of this work. These are the couple of papers we have published in last couple of years during my focus on this project. This is the DBT. We have completed the 2 project. This study was completed under the 2 different projects of DBT. One is DBT on inhibitor design, another DBT project on mapping of binding site of these drug and inhibitors complex. Well, in another project, my focus is on inhibitory action of certain gene using CRISPR-Cas system. So we design a CRISPR-Cas in our lab. So we are the first to design a CRISPR-Cas system against the biofilms. And probably if you Google the papers in PubMed, you find this is the first paper we published in Frontier Infection Cellular Microbiology, probably 2017, if I'm correct. Yes, where we have shown how you can reduce the CRISPR-Cas using CRISPR-Cas. So we designed the CRISPR-Cas in our lab by designing sgRNA and dCas9 against the target sequences and these gene separations. So we have focused on CSGD genes, which are involved in the biofilm formations and amyloid formation, and then we focus on the biofilm reduction, some virulence factors, and some pili genes, and some sun genes, and a lot of genes which are directly involved. So we design the CRISPR-Cas systems and we clone them, and after cloning, we have the vectors to be transferred into this into these cells to reduce it in E. coli model system. And there's a lot of experiments were performed. This is the one of the scanning electron microscopy photograph has shown that how the piles, see this is the pile, if you can see, can be reduced in the treated cells. The control cell, you got a lot of pile, and this is the amyloid, and this is the biofilm structure in the microstructural biofilm, which has been reduced totally in case of treated cells using CRISPR-Cas engineered bacteria. This is another pictures where we have shown the fimbriae. See, the fimbriae can be reduced. So this is the model we have shown how the fimbriae can be reduced. FIM1, FIM1 genes. The whole cascade was abolished by using this, and the work was well published in Frontier Immunology in 17 and Cellular Infection Microbiology and Journal of Biological Sciences. Well, this is my group of 17, 18 group. Where I think most of the students have already gone. He has gone to Mayo Clinic. He has joined Pittsburgh. He's in Iraq. She's the postdoc with me, and she has just completed looking the postdoc. She has got a position in USA, and these are all girls are working in my lab and different aspects. Now, well, in another project which probably we have successfully patented also this PDT technique, where we have shown I'm not going to talk about more science on the PDT, but just to give you a glimpse of it, how does the PDT basically work against the infection where even the antibiotics have failed to work. And this is how the work has been well taken in the different published in different papers, and where we have shown the laser technology, and through this laser technology we have shown so here we have developed an infection model, and this infection model was cured by PDT. This is a diabetic foot ulcer and was totally cured by a laser technique using the formula successfully shown that yes, they have successfully treated, and the work was well taken and published in top journals and core PDT journals. And recently we got a scientific report papers in 2021 on the same. Yeah, so this is what we have basically worked on, and we have developed a caries model using certain techniques in the lab and in the rat tooth. And here we have shown that how the caries model is completely cured off using the nano particles and nanomaterials. So this is what we have proved, that the biofilm, which is initially on the tooth plaque, were treated with the nanocomposites, and we disperse it by interacting with these surfaces. And this is how we would be able to cure. So this is the control the data where we have shown through OD crystal violet absorption that how the completely lost dye over here, and that shows we have would be able to remove the plaques using the nanomaterial. So this is then fortunately I got a chance under the INSA fellowship in one of the top laboratories of the country, Academia Sinica in Taiwan. And Dr. Su Ling is basically, she's again a very renowned lady. And at that time she's superannuated, and she was director of the center. She invited me under this INSA visiting fellowship program, where we have, although it was a 15 days program, but we learned a lot and exposed to the different technologies and techniques in the biofilm. And there we basically shown how we can correlate the biofilms and what we've already done in vitro into the antimicrobial resistance. Now, coming back to my own focus on AMR, having a lot of news and in different print and electronic media, and that's all news came out from the different agencies, like after the deliberations in WHO, CDC, and several papers published in top journals like Lancet and MG, where they have shown that how antibiotic resistance is a crisis, become a crisis in the globally. See, the drug resistance, 2 out of 3 healthy Indians is common. So these are all reports. So almost 10,000 fake antibiotics vials recovered from the lab. So all these things are basically the factors are reasons behind the spreading the drug resistance in our country and abroad. Now, having a look at this slide, this is the slide of CDC and WHO recommendation, and here the WHO has recommended that antimicrobial resistance in 2050 become one of the wild state of the country. It's just like a pandemic, and people will die every year. 10 million people will die every year by 2050, and this is much higher than even the death caused by cancer, cholera, diabetes, and other diseases. That's an alarming signal for us and for physicians and healthcare workers that how to handle the situation and how to understand the whole, problems behind the AMR and what are the possible reasons that we have to look into and do priority research area. Basically, they have declared as in 2014. In 2014, I was the one who discovered NDM4 superbug, and that's named as a Aligarh superbug. And that is the NDM4, the first variant in India we identified 2014. And, almost, more than 3 dozens of dailies and, print and electronic media covered that news, even international print media, international televisions, and recently Neona Film make a documentary on my NDM4 story. It's a short documentary, is available. And then Benjamin Plackett is a freelance journalist. He works on science journals, science stories, and from the Nature. And he published my interview in July 2020 in Nature. That was a big recognition for me that my interview and my quote was published in Nature. The reason behind that, because I've been working for decades on this area, and this is the comprehensive review article that we studied in Indian conditions, and we found that India and subcontinents are one of the most important reservoir of spreading the different markers of resistance in the paper published in BMC Microbiology in 17. But after the discovery of the MCR1, that is published in Lancet Infectious Diseases from one of the Chinese group, was one of the hopeless. Researchers and physicians become hopeless because no drug is left for us to treat the infection caused by such kind of drug-resistant strain, because colicin was the last drug. Polymyxin was the last drug left, and still more than 30% patients are become resistant against colicin and polymyxin. So that's one of the issue we have to look into. Now look at this, the labs in the area of Indian states and cities are working and making certain laboratories under the guidance of National Center for Disease Control. We call them National Antimicrobial Research Network laboratories working on this aspect, and ours is one of them. If you look at the 630 institutions, we have got a 4th place in the India working in the area of AMR, looking at different parameters, both human, environment, animals, and diagnostic part in one health care system. So that report was submitted by DBT Government of India in Newton's function in Delhi long back, 2015-16. Keeping that same in view, we have basically focused on several aspects of AMR. And since hospital is one of the advantage of our university, that we have a hospital inside, so we got a chance to collect several samples. So in this study, we focused on neonates, that is the babies which are just born for a day, 2, and for not more than a month. And they've been kept in the incubators and intubates them because of some ill effects like a pneumonia, or they might be a preterm babies. So what we start working on it, and we just collected the sample of Neener's, gut samples are fecal samples, and these samples were taken, stool samples were taken and to analyze for sequencing. And surprisingly we identified all these 4 NDM markers, and basically these 4 NDM markers are, NDM 1 and 4 was very deleterious. 5 and 7 is also very important markers, where causing a lot of resistance, put a resistance into the bacteria. So if you look at the series of a bacteria, we found E. coli, Klebsiella, all these common bacteria, which are, we call them as a non-pathogenic bacteria, but somehow they become opportunistic pathogens. And these all bacteria are having these markers. It's not a good sign. And if you look at the spread, you find the NF4, NF5, and NF5 is maximum in Escherichia coli, and then NF4 is more in Klebsiella pneumonia. So they have been worked. All these isolates were basically from the neonates, and we have been working on it for a couple of years, and we got published this into Frontier Microbiology. Then we have another very interesting study on the same neonates of the different time period, where we found that NDMs 4, 5, and 7 were the major cause of the epidemic. And that was the isolates taken from the neonates when there's a lot of death reported from the neonates. And these neonates which have been born for a day or 2 or 3, the survival rate was reducing in the ICU, NICU. And so we have been contacted for that, and we collected the samples and test them, and we thoroughly analyzed them, and we found these 3 more strains which are epidemic there, and they were all from the same group and class. And if you look at the MIC, the doctors was giving the medicine, all these medicine, imipenem, meropenem, ceftazidime, cephalosporin, 3rd, 4th, 5th, 6th generation of antibiotics. But you see the MIC, I mean, those who are in the area, they can understand, 1024, this is a huge amount. And if you look the concentration of a drug being given in a normal person should be 1 microgram per ml. And this is almost 1,000-fold high MIC. I mean, that's amazing. So, I mean, nobody can give 1,000 microgram MIC to kill the bacteria, to cure the infection. Otherwise, you will kill the person. You will all spoil the kidney. The person should come on the dialysis on this very second day. So that's the situation. So we got a sequence, and we identify the reason behind and what are the basically markers in this strains, and we have got this study published in one of the core journal, Microbial Drug Resistance. Now later on, we keep going on, and we identified a very interesting strain, Seperia lepagi is basically very, very rare strains, generally not found, but in an ICU I got this strain. Unfortunately, or fortunately from my perspective, we got a rare strain, but unfortunately from the NICU perspective. This Sedecia lepagi is basically a strain showing very high MIC, and kids are being affected, neonates being affected. So we again get a sequence and found the gene involved in it, and what are the different associated markers. So we found the NDM was there. And again, NDM was creating a problem by hydrolyzing all these drugs, and these drugs are no more be effective against the NDM producing these bacteria. And so we have identified this, published in IJA core journals. And again, in another study, we characterized the Klebsiella pneumonia and ICU hospital strains again, and NDM1, a lot of NDM1 was found in it, and that study was again published here. And this is a very interesting story of one patient admitted into the hospital, and he was UTI patients, and the drug resistance was found in uropathogenic bacteria, and these UTI patient was recurrence of UTI was there. And so what we did, we just took a strain and we found what are the basis of not recovering. So we found NDM 6 was one of them, but besides NDM 6, since we've got a sequence of a whole genome along with the plasmid in it, these are all strains present on the plasmid. These are all markers present on the plasmid. So what we found, that very interesting thing, that we have got a multi-resistant marker, series of markers in the genomic DNA as well as in this, through different transposons. And these transposons are transposable element, which can cause a jumping gene from one strain to another strain. And that's one of the reasons the strain was and that patient unfortunately would die after a few days after the kidney failure. Now, keeping that all in mind, we thought of moving towards more understanding the mechanism. And I'm not going to talk about the different mechanism, but just focus on what we are working on, the plasmids and conjugal transformation and transduction of these bacterial genetic markers. So I just explained you the mechanism, basic mechanism. So this is the beta-lactamase ring, and this is the beta-lactamase enzyme. Beta-lactamase enzyme is, I have not having a slide here, but these are the enzymes which are like NDM is one of the enzymes, CTXM is another enzyme. So these are all produced by the bacteria. What they do, they just hydrolyze this to this chemistry. So this is the chemistry behind hydrolysis of this ring, beta-lactam ring, which is the intact nucleus of an antibiotic. And this basically antibiotic is causing the killing the bacteria. So when you kill this antibiotic by this enzyme like this, you open it, then antibiotic becomes fail to work. And so this is the reason of resistance. So what we are focusing on, looking the different sites here to understand and to control by designing several drug molecules on it. And these drug molecules can be in future, can be act as a synergistic effect of these drug molecules are the inhibitors molecules along with the available antibiotics. For example, if you have clavinate. So clavinate is inhibitor work well in amoxicillin. So amoxicillin alone will not work, but amoxicillin with the clavinic acid will work, and this all together will definitely reduce the resistance. Now, so first we have focused on one of the very interesting. This is the NDM1 structure. All these enzymes, and this is basically the position of TRP993, which is not in the active site. This is beyond the active site. But what we thought of, even beyond, even distant site mutations, a distant site residue also play a role. So we are the first to start working on distant site mutations and looking into, so we make a dozen of mutants. This is one of the example of mutant we have made, TRP19, and we replace with alanine. And then we have shown the loss of function of the whole enzyme. So what we have shown, that we have cloned this enzyme, this gene expressing this enzyme, having the mutation, and with the control, and we found thus this enzyme is lost its function. So it means that very residues, TRP93, is very critical in making the enzyme more effective against the antibiotic. And that published in Antimicrobial Agent Chemotherapy in 2015. Well, after that, we have got several papers. In 2017, we have got this potential inhibitors, again, NDM1, then we can have several other inhibitors. And recently we have got a paper, we have got a paper in ACS Omega, which we have not shown probably here, where we have shown, how basically how the broad spectrum inhibitors work. And so we are working on non-beta-lactam broad spectrum inhibitors, which can work against these inhibitors. So this is all about a glimpse of my research for the last 20 years, my journey, and which we have finished, I think, within the time. This is my group. I think some of them, Wockkardt has already left. Wockkardt has gone to Mayo Clinic. Abid is in AIMS, I think, with Dr. Shujata. And Abid is in Pittsburgh, and she has just finished the PhD thesis, and she's got a paper in scientific report recently, and she has developed a technology on DFU. Aisha is working on a very interesting pathways, which we are very exciting to look into and work on. The first time we have explored the Pseudomonas biofilm pathway. Shamsi is working on epidemiology. She's a postdoc. Lama is working on very interesting applied aspect of PDT, where we have worked on earlier as well. Some of the students are not here. I'm sorry not to acknowledge, but I think we have a group of a large number of students which already have gone, and we have shown the data from their papers. So these are some of my grants, earlier grants based on that work, depends on my collaborators, Zarili and NICU, Dr. Manazir, Ashok Sharma from CNAP, and Praveen Verma, and IPGR. So these are some of my collaborators, which we keep interacting with some facilities and support in order to attain this work. With this, I thank you very much. And again, if you have any queries and questions, you're most welcome.
Absolutely. Thank you so much, Professor Khan. This episode was, I could significantly see how research has gone forward, how your work is contributing not only on a regional level, but it is going global. It is putting science, and it is putting the advancement on the map of how the country is also doing well. And through your research, I am sure it's going to go further ahead. So thank you so much. Would you like to say something before I close?
I think this is all. I think I thanks a lot your platform of Biofootprint. who's inviting people like me. I'm not a high stature person, but I'm a very small researcher, and I'm still learning and a student. But my whole focus is that always I'm trying to learn some new thing. And when you look my research, I started from A and I'm trying to go to the Z, trying to increase my research profile, and trying to increase every paper came from my lab, and every thesis came from my lab. So I've so far guided 25 PhD thesis. When you go my first thesis and we go my 25th thesis, you will find a vast and great change in difference. So this is what my always focus, keep going up and up, and thinking always beyond your imagination, and basically always work and think towards the beyond the comfort zone. So if you keep yourself in the comfort zone, you can never rise like anything. But if you go beyond comfort zone, you can always touch the stars in the sky. Thank you very much.
That's a very beautiful thing to say. And on behalf of Biofootprints, I would like to again formally thank you for giving us your time. And I'm sure a lot of people are going to watch this and take a lot of it from this episode. So big thank you. Thank you so much.